The overall goal of this procedure is to synthesize a micellar enzyme immobilization and stabilization polymer. To accomplish this, first combine the Nafion suspension with the alkyl ammonium bromide salt. And vortex the mixture in order to exchange the protonated form of Nafion with ammonium salts. The second step is to dry the resulting polymer by pouring the mixture into a weigh boat and allowing the solvents to evaporate. Next, soak the polymer in deionized water for 12 to 24 hours to remove excess salt. After salt extraction, rinse the polymer with water. And then allow the polymer to dry completely by evaporation. The final step is to transfer the polymer to a clean vial and resuspend it in solvent for use in enzyme immobilization. Ultimately, this micellar enzyme immobilization polymer is used to improve the stability of enzymes in activity assays.
Generally, individuals new to this method will struggle because this procedure seems extremely straightforward, but the drying procedure is critical to the fabrication of a functional polymer. Demonstrating the procedure will be Shuai, a graduate student in my laboratory.
To begin the procedure for modifying Nafion with quaternary ammonium salts, measure a 3-fold molar excess relative to the sulfonic acid groups on the Nafion polymer of the